There are multiple ways to keep in touch with the Wolf Connection podcast. Through our Instagram handle, the Wolf Connection Pod. And for comments and questions, send us an email to podcast@wolfconnection.org with your comments, questions, and guest ideas for Stephen and myself. You may hear your question answered on an upcoming podcast. Thank you for your support, and howls to you all. Welcome to the Wolf Connection podcast. I'm your host, John Kalfa. Let's talk about some moles. Joining us from Princeton, New Jersey, she is the Associate Professor of Ecology and Evolutionary Biology at Princeton University since 2019, when she got her promotion from being an assistant professor. This is Bridgett VonHoldt. Bridgett, awesome for you to make the time and come on here with Stephen and I. How are you doing?
Hi. Oh, I'm so excited. I'm thrilled to be here.
You had said in your, I know, we're thrilled. It's a lot of, you remind me of our podcast we had with Ellen Brandel, who studied a lot of really genes and things of that vein. So this is, I did a lot of reading. Yeah, heavy science. So this is going to be a lot of explanation from you, but we are really excited to get to have you on here to describe the work that you're doing. So just give us a little bit of a background, where you grew up, why evolutionary biology and things like that, and how you got started at Princeton.
Sure. I'm the product of a military family. My dad was in the Air Force. So from the age of 3, we moved around. So I don't really have a hometown. I spent about 5 years in every location. And as I graduated high school and entered college, I continued on with that every 4 or 5 years moving. So I was born in Iowa along with my identical twin sister.
Oh, wow, cool.
So that was the closest hospital because we lived in Minnesota. It was a small town. It's still a small town. And from Minnesota, we moved around to Florida and to Colorado and overseas. And it just keeps going. Now, the field of study, why evolution? This really started off from a very young age. I think many people who are in biology have memories of, I love being outside, I love animals, and then being concerned with endangered species and loss of habitat. Those kinds of commercials and adverts always got me. And I really wanted to do something to help create a healthier place for our animals and wildlife. So I was always interested in trying to help animals. And that kind of came through as internships with animal shelters or vet clinics. And eventually I stepped it up and I worked in a zoo for a couple summers. And I had a lot of public outreach. It was kind of great to talk to people about the biology of animals, where they live, why do they do the things that they do. And that kind of led me to thinking about a biology degree. Now, I started off, I think you already noted, I started off my college experience as a psychology major. And, that was kind of a hard decision because I loved biology. Psychology was really incredible, thinking about brains and cognition. That lend itself to me learning a lot of statistics. And I liked that. I found that actually really fun. And I merged that with biology as I was continuing on in my education. And one thing led to another. I learned how I could merge wildlife and conservation with a very sort of quantitative and sometimes challenging field of focus, which is genetics. So I've loved it. That has been very good to me. And it continues to push me every day to think about new things and new ways of solving problems.
Yeah, just to give everybody who's listening a little bit of a background on, and if I mispronounce something at any point, please correct me. But I believe it's pronounced genomics,
I believe,
of hybrid zones. And genomics, thank you, of hybrid zones and ecological epigenetics. So what are those?
That's right.
Good. All right. I was 1 for 2. Give everyone just a breakdown, I guess, in somewhat layman's terms of what those two fields of study are and how you factor into the studies that you're doing with those two focuses?
Great. I'm going to start with the latter because I think that's a little more contained and kind of separate from the hybrid zones. So starting with the ecological epigenetics, I became interested in this because I had spent all my time thinking about genes, but now I wanted to understand when genes don't have the answer, there is something else that might. So we can think about in a single day of our lives, how we wake up in the morning, we have something to eat, our genes are now producing, they've been stimulated to produce the proteins that will help us digest our meals. And when we're done eating, and as we're still digesting, we're making those proteins. But when our food is done and processed, we don't need those digestive proteins hanging around anymore. So our genes turn off, and we don't need those proteins until we eat again. So that means even though we always have the genes for those proteins, that gene is not always turned on. There's a time and a place for genes to do the things that they are programmed to produce. And that's essentially the epigenetic part of it. I've thought about genes and thinking about mutations, but now I wanted to think when you have a gene, just because you have that DNA doesn't mean it's always on or it's always off, or that it's on at the right time and the right place. When we apply this epigenetic concept to ecology, we begin to ask the question, for example, if I take a turtle who lives in the desert and I completely isolate it in a rainforest, it can't get back to the desert. I've removed that possibility. It has the genome, the set of genes that has evolved in a desert environment. But now the same set of genes in that turtle live in a rainforest. And we want to know to what extent can this turtle actually manage a totally new environment with a totally new set of pressure, different rainfall, different temperatures, different competitors, different predators. And some of the genes might actually turn on and off in new ways that help the turtle deal with a totally new environment.
So, and I assume this concept of it sounds like this concept of genes going from inactive to active, in use to out of use, is sort of is sort of fundamental to this study, to this arena of science. How is that? How are these things observed? Like, how do you observe a gene turning on and off? How is it documented?
That's a really great question. There are a couple different ways that we can use sequencing technique to know if a gene is turned on or if a gene is turned off. One of those things, actually, there's more than just two ways, but the two most common ways in my field of genomics is to sequence the little product that a gene makes. Right before it turns into a protein, it just makes a strand called messenger RNA. And that's the little molecule that turns into or it constructs the protein. So if you go through an organism, let's say the turtle that lives in the desert, you can sequence all of its messenger RNA, take a little blood draw, maybe in the midday. And at that point in time, you will know all of the genes that have produced this messenger RNA. Those genes are on. If you now sample the turtle living in the rainforest and you do the same process, you'll know which genes are turned on. And we can simply compare the two lists of genes. Are the same genes always on? Are there different composition? The other way, the epigenetic way that I use is sequencing for just a little chemical marker that attaches to DNA. When that marker is attached to DNA, we generally presume that that is an off switch. So this is called a methyl group. It's a tiny molecule of a carbon with 3 hydrogens. And when that is stuck onto DNA, it acts as a local stop sign that anything in the vicinity, we're presuming as a universal rule, that when the methyl group is there, the neighborhood of genes are shut down. So I can sequence that in a slightly different way, but still get lists of genes that are maybe methylated and turned off, or unmethylated, and thereby we presume they are on.
Does it seem like every individual of a given species behaves the same way in terms of their genes when they turn these genes on or off?
There are a handful of these universal housekeeping genes that you'll always need to have this thing produced in this tissue type or at this time based on the physiology and what the animal is experiencing. And that can be specific to mammals or to vertebrates or to egg-laying species. And then what's really interesting is when you kind of remove all of that, because everybody will have that, and you start looking for where are their unique differences. And that tells us something about perhaps immediate response to some sort of external stimuli. And we can try to use that information to investigate if this could be an adaptation in the making.
Is there a condition, or I don't use the word disease because I can't say that it's negative necessarily, but is there a condition in which, in your field, that would describe a scenario in which an individual of a certain species should be using a gene in a certain way, but it's not functioning properly?
Yes. So there have been lots of studies in the past several decades looking at humans specific now, I'm going to talk about humans, that when there are major catastrophes or stressors in someone's life, that leaves an epigenetic imprint on their genome, and it changes the way that their genes are expressed.
Oh, that's fascinating.
And if you think about, I forget what these book names are, but there's usually a focus on like really incredible athletes who push their bodies to the extreme. And then also major cultural disasters like concentration camps or famines. The level of stress that those individuals experience leaves a mark on their genome. And what we're learning is that even though it might be helpful in the time of stress, some of those markers get inherited to their children and to their grandchildren. And yet those markers are helpful in, let's say, a super athlete. But if their grandchild is not an athlete, their body, though, their genes are functioning like a super athlete. So those are called mismatches. And that means that if your genome is acting in one way and it doesn't match what you're physiologically demanding from your genome, that could result in maybe some metabolic deficiencies, maybe some developmental abnormalities, or just differences from what the expected routine is. So yes, it's a really interesting field. I think people are just starting to learn how to really get at these questions. And for me, using the techniques in wildlife helped me and my colleagues ask questions about, let's say, urban pollution versus natural pristine landscapes. If a bird, for example, can fly from a protected landscape into the downtown area of some heavily urbanized area, those are two different environments, but it's the same bird. And we want to know, how do the genes respond? Are they acting in the same way? Are the genes acting differently? Which genes act differently? And that tells us about what the bird is experiencing and maybe what could be considered a stress.
Yeah, it is. That's crazy. So when you're saying too, with the, let's just say the super athlete, it's what you're saying is there is basically a deficiency if their kin are not using the genome or the gene the way it should be used as their parents or grandparents used it. It can essentially be a hindrance to them. Am I hearing that right? Is that kind of how that works?
Right.
Okay.
Right. So for famine, this has been studied in the aspect that maybe our great grandparents experienced severe famine and had severe limitation of calorie intake, quality of food, quantity of food, and their genes responded to help them conserve everything they could. Metabolism changed, and those who survived had basically the right sets of genes going on and off in the right way to deal with famine. But now, when they survived and they had children, and then those children had children, we can now imagine that there's a genome that has the imprint of famine, but the grandchildren are not experiencing famine. And there's been a handful of studies that show that these grandchildren seem to have perhaps a non-random increase of metabolic disorders.
So really the outside factors are what's influencing the inside factors of our bodies and our genetics. That's why.
Yes, this is what
so it's
epigenetics,
something that's beyond our control, basically. Yeah.
Yeah. But not everything will have that inheritance. And this is still, a huge question in the field. How much of that epigenetic change is actually transmitted to our offspring? Some of it is short term. It will be erased as, very quickly after it has been set down and been used. And other times it's far more permanent. And we really want to know, A, what are those changes? Just strictly, what are the changes? And then B, of those changes, how many of those are permanent 2, 3, 4, 5 generations later? Because that will help us think about this in an evolutionary context, that now there's a change that is longer lived, and we need to understand the consequences of that.
Absolutely. So when you're studying canids, because this seems like such a great bridge here, how does this factor into the domestication of really wolves, wild wolves coming into now the domesticated species that we have? I don't know the number, but clearly there's thousands of different species of domesticated dog. How do we get from the wild cousin down to this? Yeah.
I'm going to make a very polite correction.
Go ahead, please.
Dog breeds are not species, they're breeds. All breeds belong to one species, and that's the domestic dog. And this is a very common sort of misphrasing by people. And I'd love to have a record for everyone to like, oh yes, it's just a breed. Yes, so that connection, so epigenetics, in all honesty, I have not used it very much in canids. I use epigenetics for very specific questions when I think about dogs compared to wolves. So let me give you that kind of example, and that will cover the domestication history of dogs.
Sure.
I have spent lots of years thinking about how we have changed a wild wolf into a dog. And a lot of people think about that. And there's a lot of research that catalogs changes, whether it's the way an animal looks, the way it sounds, the way it behaves, how often do they reproduce, how quickly do they develop, lifespan, all of that stuff. I come in this area from the genetic side of it. So this is a natural experiment. Well, by people. So it's an experiment that has already happened. People have completed this experiment, and I don't need to create a dog colony in lab and do some breeding. It's already been done. And we have really good records of it too. It's not a complete set of records, but we do know generally where and when dog breeds were created. So over time, my questions have been, how have we changed the genome of the gray wolf into the set of mutations and genetic differences that have created a dog? And not just one dog, but like the over 400 breeds of dog. And they're all very different. They can be from each other. I started off, this was during my PhD, where I worked with Dr. Robert Wayne at UCLA. and I had a committee of lots of other dog people. And we essentially found that when we compared gray wolves with a collection of dogs, we kind of grouped them all together because we want to know all of the genes that represent dogdom and compare them to all of the genes that we find in gray wolves. And some of the major differences in the genes had a lot to do with the way a dog's skeleton is proportioned. So growth type genes, genes that control fur color and structure. So wire-haired, curly-haired, the color of the fur. And some of the genes that interested me were the genes that seemed to be kind of connected to a possible question of change in behavior. Now, I don't formally study behavior, but we do know that a dog behaves generally very different from a wolf. We're not going to approach a wild wolf. In fact, the wolf will run away from us. And our dogs have a wide range of being a leech and being kind of aloof, but still, very close to one of their humans, if not maybe both of their humans.
Permanent puppies.
Yeah, right. And I've sort of stumbled upon some of the genes that seem to influence that behavioral difference. Now, when I looked into it further, and I've continued to investigate this in my career, that what we're finding is a handful of about 30 genes that seems to get disrupted in their regular expression pattern. So thinking about epigenetics, we expect this handful of genes to express in a certain way, at a certain level, at a certain time. And we don't know all of the answers yet in dogs, but what we do know is that those genes in dogs are expressed very differently than those same genes in wolves. And the way that those genes are expressed differently is influencing how social the dog is or the wolf is with people. Now, these 30 genes are well described in people. This is actually a handful of genes that when they are mutated in people, it results in a syndrome, a behavioral syndrome called Williams-Beuren syndrome. Now, Williams-Beuren syndrome is one of the ways that it can be diagnosed is through a variety of behavioral surveys and questionnaires. But essentially, Williams-Beuren syndrome is characterized as people who lack stranger danger. They are friends with everyone. In fact, that's one of the slogans is everyone in the world is my friend. If you have Williams-Beuren syndrome, there is this social butterfly. You are distracted by socializing with people. Now, there are other aspects of the syndrome that are not nearly as lighthearted. There's several biomedical conditions. But it's the behavioral aspect that really struck me because it wasn't hard to imagine there are dogs like this. There are dogs that don't care if they know you. They already love you. They loved you before they met you. And these genes now had a story that really enticed me. We know what they do in humans, and it's related to social behavior. And now I can investigate those genes in dogs and compare them to wolves. We also can compare those genes and how they're expressed in wolves that have been raised in captivity by humans. Because there's another expectation that, should you get a wolf pup at a really, really young age, we could socialize it. And it could maybe be just like a dog, but it doesn't have the genes expressing in the same way as the dog. So the field has suggested that you can bring the wolf so far close to a dog, but very rarely will you get that wolf being a dog, basically.
All right, so you mentioned dogs and wolves, but did you happen to address the issue of kind of like hybrid wolf dogs and how their genes manifest?
Yeah, so I've got a tiny story on that with respect to coyotes. Coyotes don't have these genetic changes, by the way. I've not found any of these, hypersocial genetic changes in coyotes, except, as you've mentioned, Stephen, for coyotes that show a tremendous amount of dog ancestry. Like, for example, they're either a first generation hybrid or a second generation that's been back crossed. And we just put a paper up on bioRxiv 2 days ago, so I can send you the link if you want to also promote that.
Of course.
But what we found is that, anecdotally, there was a handful of coyotes that were being a little too willing to accept handouts from people in Queens, New York. And, this creates a lot of concern, both for human safety and the welfare of the animals. There had, from what I believe, all of the animals had been euthanized since this was a public danger risk. But in doing the genetic analyses, we can have some explanation why they were so tolerant of living in a densely populated, neighborhood at a location where people could feed them. And it's because those hybrids, coyotes, when they carried their dog genetic ancestry, they carried these hypersocial mutations. And those are the mutations that shape the expression of the genes that lead to more interest in people. So in understanding the ancestry makeup of an animal will help you understand if the animal will have tendencies to behave more dog-like or more wolf-like.
So we're really looking at, especially coming from our perspective as a sanctuary that rescues wolves and wolf hybrids, really, you don't know what you're getting because there's so much crossbreeding. And so you
right.
Like you said, there's wild, basically every pup that's born is a wild card because you don't know what's going to come out.
Even within the same litter, depending on Mendelian inheritance, there could be two pups from the same litter that have completely different mutations that they inherited from mom and dad. And you can get different behaviors from full siblings. So I'll offer up, if you ever want to genotype your animals or anyone out there, get in touch with me. I'd be thrilled to tell you the genotype content of that animal with respect to these behavioral mutations.
All right, so let me ask, is there any consideration gone into inbreeding as it relates to this concept?
You might have to unpack that for me in terms of how the behavior?
In terms of how their genes behave.
So, right. Dogs generally, breed registered dogs are generally quite inbred. And what this means, and I know that there's often a negative feeling when it comes to that, what this means genetically is that more dogs have the same, like the exact same genetics. They've inherited the same fragment. You and I will have the same genes, but we've inherited them from totally different lineages. Dogs will inherit them from the common sire or the same breeding pool that just pumps out puppies, 3 times a year or something. So because everyone is genetically the same and the same lineage, that the genetic variation will be very limited. But what this means for the behavioral mutations is that there's an expectation. I mean, you could go through theory and say if everyone has the same genes and everyone has 2 copies of these mutations, then everyone else should have 2 copies because that's all you have in the gene pool that's breeding together. But what's curious is that this is actually a part of the dog genome where there's a tremendous amount of variation. And I don't know why. I don't have that answer. But I have found, and I've genotyped 3,000 dogs and wild animals. And nowhere do we find that a breed, name a dog breed, is absolutely fixed and identical for the set of mutations. So I can't say all boxers have 5 mutations. We could find a boxer with 0 mutations in these behavioral genes and a boxer that might have 8. The variation is still there. So there might, as an evolutionary biologist, I would hypothesize something is maintaining that variation. On the flip side, I also talk about these mutations as possibly being part of a dog's personality. Depending on how you define personality, we do know that even 2 pups from the same litter could have dramatically different personalities. And I have found, because these mutations are so variable, that probably this has some role in helping that dog sort of become its personality.
I see. So a female dog can get pregnant by 2 males. Is that true?
2 male dogs can copulate. I think it's more common in dogs to find extra 2 different fathers within the same litter. We've searched for that in wolves, and I don't have any evidence of that in wolves, but I know that there have been records in dogs. It's not super common from what I know, but I don't scour that literature too frequently.
It is true, man. You can have 6. If you have several puppies in a litter, they could look different in every way.
They could,
of course,
but it could also be the way gene combinations interact with each other. So if dad and mom both have completely different types of genes, these would be called alleles. Then when you put those alleles together, there's many different ways in which you can match them up, and they get inherited in a puppy. That every puppy, if you have 6 pups in a litter, they could all maybe have very striking differences, but that doesn't always mean that there are more than one, father's genetics in there.
Okay, so even in examples where there is generational inbreeding, kind of, and that's obviously how we get purebred dogs, and that's why a lot of them have the same issues,
right,
litter after litter.
Right.
So it's not exactly proportionate in terms of the narrower the genetic bottleneck, the more often the same mutations manifest throughout individuals, and the more genetic diversity, the more diversity you might see in their genetic behavior. There's kind of like a gray area where even in cases of a narrow genetic bottleneck, there still seems to be diversity.
Yeah, let me try an analogy and see if it works. If we imagine a giant bowl or a bucket of marbles, and let's say there's 700 marbles in there, or maybe we should be friendly to the non-math inclined.
Okay.
I can't do math on the fly either, so
okay.
Let's do 1,000, because that's a well-rounded number.
There you go.
1,000 marbles.
Good job.
And each one of those marbles is different. They're just all different.
Yeah.
So we have a tremendous amount of variation. It's the maximum we could have. And that's the early pre-dog, proto-dog thing. That's the ancestor. Now, when humans became focused on creating all of these fancy breeds, and this happened a couple, 200, 300 years ago, we took that bucket of 1,000 marbles, and everyone in the neighborhood basically would say, this is what I want to see in a dog. And they would maybe grab 50 marbles. And that's what they started with. And everyone grabbed 50 marbles. And now with those 50 marbles, those are the ones that I'm breeding with each other. And I make more baby marbles, but only from that 50 that I grabbed.
50, right.
Now, every now and then, if I'm like, my dogs are all like, they're not doing well, maybe they're really inbred, I need to do something to help, I'm going to ask for like 2 marbles from way over there, because that other little population, I like the way their dogs look. Mm-hmm. It seems to be a good match. So I'll say, hey, can I just borrow 2 of your marbles? And then I mix them in with mine, and I've boosted just a little bit the genetic diversity. But still, then I keep those 52 marbles together for another 100 years. And now every pot of marbles in and of itself is limited in diversity and is inbred. But if you look across all of the buckets of marbles, you have tremendous genetic diversity because we started with 1,000 different marbles. So dogs are mostly devoid of genetic diversity, except when you look across all of the breeds, all of the genes of those breeds. And when you start mixing breeds, then you get a lot of new things combining, and you get a new, designer dogs, right? You get doodles everywhere. So that's kind of how things work. I hope that was an okay analogy.
Yeah. No, that's good. Yeah. That's good. It seems like it's the same that what happened with the wolves in the wild when the black phase gene found its way into the population, because Leo was the one that turned us on to this, Stephen, Leo Leckie, was that once that gene entered the pool, it diversified so much, because you were just talking about grays and timbers mating together in the wild, and now you added this extra gene that not only enhanced, or like you said, they started with, let's say, 1,000, and that almost seemed like it was almost another 1,000, because it helped them not only with coat color and fighting off disease, it really diversified the gene pool. And it almost seems that any pack of wolves you see, at least in the lower 48, has some variation of that black phase gene that's in the pool, as opposed to just seeing all whites and grays.
John, I'm so impressed with your research. Yes, I was a part of the team that discovered the mutation in wolves. And
oh, cool.
Yeah, it was led by Greg Barsh and Tovi Anderson when Greg was at Stanford. And this was several years ago. I think we published it in 2009. And there was an indication. The reason we looked in wild canids is because in dogs, there are several genetic ways to get a melanistic dog. A golden retriever or a golden Labrador is a different kind of genetic black than a poodle, a black poodle. They have different mutations, but it all leads to the same switch in pigmentation type. So that came up, and Greg had come down to UCLA and told us this story about the dogs. And I was like, well, funny, you should mention this. At the time, I was building the pedigree of Yellowstone wolves. And I love pedigrees. I was putting together these animals, and I was noticing just anecdotally, like, you kind of have two melanistic wolves. When they mate, they can have both gray and black pups. And then you have two gray wolves mate, and they never have black pups. And like, I don't know, is this an X-linked mutation? Is this dominant, recessive? And I could work it out. It was just like, this is like the rare occasion that your genetics 101 class turns into real life scenario. It is a Mendelian trait, and melanism is dominant. So I could just map a pedigree, look who has the black color or the gray coat color, and the inheritance just follows beautifully. So Greg Barsh, Bob and I had told him about this. And we're like, we need to discover if this is which mutation of black this is. Is it the Labrador? Is it the poodle? Or is it something different? So that's when the K locus, this is a 3-base pair deletion in a gene called canine beta defensin 103. This is a gene that has normally a function in immune systems. So it is an antimicrobial gene. It helps your immunity spot microbes and defend the animal. It's normally found expressed on the skin. This is where microbes will live generally, the first defense. But when the mutation happens in this gene, it creates this different protein called a ligand, and that protein binds the pigment switching from yellow to black. So we know why an animal can be black, but what we were curious about is, what are the other consequences? Are black wolves living longer? Are they healthier? Do they have some sort of advantage over the gray animals who don't have this mutation? But you are right, John. This is a new phenotype that came into the wolf genome from a domestic dog, from breeding around 14,000 years ago. And Raina Schweitzer, who's a postdoc, she was a postdoc at University of Montana. Now she's doing a postdoc at USDA. But some of her research has found and described, sort of pinpointed when this K locus mutation entered into the wolves, how it spread across geography, and how it really can play a role in describing phenotypic diversity. You have black and gray, along with like the white and the brownish and the blondish kind of wolves. But also we can look at how old those mutations are across a landscape. And it's just fascinating.
So when you're looking at it, when you're diagnosing this type of effect of what's going on, and I want to hop into the red wolf because again, I feel like this is a good segue here, because we're discussing about coyotes, and the article that I read was really fascinating in describing that it seems as though, and for those of you, and I'll have this article posted, or we'll have the article posted in the description as well, along with the papers, so you guys can read along and understand what we're getting into, is that it was almost this ghost gene that seemed to be, I don't know whether it was packed away, and that's what I guess that's what it seemed like, and obviously you can go into it, but it was really a ghost allele that was found in these trapped coyotes of, again, I'm going to say repressed, I don't know if that's accurate or not, that red wolves and coyotes were essentially crossbreeding or cross mating, and that the line between whether it was a coyote or a red wolf was almost in question. Yeah, because there were some that were found, I believe what I read was that it was up to 69% red wolf. So it's essentially, if you're looking at numbers, it's a red wolf as opposed to a coyote. Yeah, exactly.
I'll quote you on that.
Yeah. So just go into how this was all found, and this was down in Louisiana where they found these coyotes and these crossbreeds.
Yes, I'm going to give you the full story if you have time.
Please go ahead. We do. Go ahead.
There was in my job, which I love, and I love when people email me because I just love the connection to anyone who finds this interesting. So from a very early age in my career, like when I published my first paper on dog domestication, I would get emails from people who own dogs, and it was just so amazing. They would say things like, my dog is amazing. You should have my dog in your study. Here's a picture of my dog. My dog is brilliant. And, not to make light of it, but I would often read the email, have these really high hopes, like maybe this is the dog that's doing taxes. And then the picture attached would be the dog is playing fetch. Like, that's great. He's a dog. Perfect. More recently, those pictures have changed into wildlife camera, like remote pictures. People put up game cams in their backyard or property. And they're like, oh, hey, I got this really weird picture. Can you tell me what it is? And I really appreciate these interactions because I also try to tell them how difficult it is to make a conclusive statement on a picture that's maybe at night or with no scale bar, no size reference. Like, it looks like it could be the butt of a canine. I don't know anything more than that because I see its butt and its tail and like, that's it. So this is a common thing for me. And 2015, another one of these email threads went around, but it was now from academics because there was a community, a citizen in Galveston who had taken some beautiful pictures, had been watching these coyotes for a long time, and was reaching out to scientists to see basically if anyone would study them. And there were a lot of comments from the academics, what it could be, maybe what it wasn't, maybe how difficult it was. And I got involved in the chain and I just said, me, I'll do it. I'm happy to do it. It was 2 samples. He had 2 roadkill animals that he took a little tissue from. And I said, yeah, just mail that to me and I'll do what I can. And his name is Ron Wooten. I just saw him last week because I visited Galveston again. And those 2 samples, we sequenced them and compared them to every living lineage of canine in North America. So I basically was doing my own embark test for wild canines and asked, are these samples, these little tissue samples, do they contain dog genetics, coyote genetics, red wolf genetics, gray wolves, eastern wolves, Mexican wolves, Great Lakes wolves? And that test returned back that these coyotes, these 2 coyotes in Galveston, an urban beach town, had upwards of 32% red wolf DNA. And I was astounded. I had no idea, although I did see the picture that Ron had been circulating, and I thought it looked like a very curious animal. So I wanted to go on that to say, well, it looks interesting, and now the DNA tells me it's interesting. We then followed up with a separate study that was funded by U.S. Fish and Wildlife, where we went to southwestern Louisiana. And we went there because this was the last geographic place where wild red wolves lived in the 1970s, which then went on to be declared extinct in the wild by 1980, the year that I was born. So I feel a kinship to understanding all we can about red wolves. They're no longer in the wild. The red wolves in North Carolina are an experimental population and they're protected. And they're down to something like 15 animals. But in southwestern Louisiana, there had also been a large swath of land that was protected either as national wildlife refuge property or privately owned citizen property. So we got permission to go to this property called Moore Odom. It's run by the Moore Odom Foundation. And we've trapped canines. This is part of the group called the Gulf Coast Canine Project. It's directed by Dr. Kristen Bresky at Michigan. And also, I'm one of the co-founders, and Dr. Joey Hinton, who's at the Wolf Conservation Center, he's also a co-founder. So Joey is he traps anything. He's phenomenal. So he went out to Southwest Louisiana, trapped 30 animals last year, 30 animals this year. But I had the DNA last year and I sequenced it. And that's where, John, we have this animal that came back at 69% red wolf. Now, there's 2 things here. The red wolf ancestry is incredibly important, but I define red wolf ancestry from the genetics of the captive breeding animals, the species survival plan of the 200 something animals that live and breed in captivity. Now, we know that those animals all came from 14 founders. And those 14 founders were captured in early 1970s from this giant effort in eastern Texas and southwestern Louisiana, where biologists wanted to save the red wolf and made a conscious decision that the only way to save them was to go out and catch the last wild ones and move them into captive breeding programs. So it was an intentional extinction, but they are extinction in the wild. In that effort, they trapped something like 400 something canines, coyotes, red wolves, coyote dogs, red wolf dogs, all of the options basically you could find. And many of the animals were disposed of. Either they were problem coyotes that had been called in, and it was indeed turned out to be a coyote. But every now and then, they came across an animal that was bigger, had more wolf-like stature, behaved more wolf-like, and they would capture it and translocate it up to Tacoma, Washington, which is where the first captive breeding program was established for red wolves. So this all came from basically the southwestern Louisiana area. The definition of red wolf is from the 14 founders, and everybody descends from there. Now, the ghost part of this, I'm finally going to get to the ghost part, is it's very easy to kind of convolute that with red wolf ancestry. We know that 69% animal, that is a red wolf definition. It has that much DNA as I identify and define it as a red wolf from the SSP animals. Now, the ghost part is because what I also do is for every animal, I can count how many genes it carries that I can't find anywhere else. It's not found in a gray wolf. It's not found in a dog. It's not found in a red wolf, an eastern wolf, a coyote. It is a genetic variant that nobody else carries. And so I'm labeling those as ghost genetics because we suspect that those are genes that survived from the ancestral population of red wolves that maybe never got captured in the 1970s. Now I'm going to follow this with an anecdote saying, how easy do you think it is to catch every coyote across, a multiple state region?
Impossible.
Not easy.
Exactly, impossible. So as much as they did trapping 300 to 400 or so animals is a huge effort over a 5 to 7 year period or 8 year period. But we suspect, and I'm sure everyone suspects, you didn't catch all of them, and maybe there's a red wolf that just was sneaky enough to never get caught. And if so, that animal would have very few other red wolves to mate with. So by nature of density, it most likely would have chosen a coyote mate, passed on its red wolf genes, and it would eventually die. And now those genes are surviving in roughly half the quantity in its offspring. Those offspring grow up, possibly mate with a coyote, and that ancestry is reduced again. But now if we have lots of those little scenarios, pockets across the multi-state region, there's a chance that a little bit of red wolf ancestry here and there can meet up, and all of a sudden an animal might have more than either of its parents. But also those are the genes that never made it into the captive breeding program. So we call them ghosts because we thought they were gone. We thought they were extinct. They're almost zombies even. They've been brought back to life because we found them. And we weren't, I don't know how often people were ever looking for them, but we happen to be in the era of genomic technology where we can do this at a large scale for a reasonable cost.
So essentially you have to retain these red wolf ghost alleles, you have to basically isolate these populations and make sure they just breed with other red wolves, because if they interbreed with too many coyotes, it's lost.
That is one idea.
Okay.
I think that, we are very sympathetic and aware that legislation to protect coyote is not going to happen. And that's not what we're asking for. That's not what I'm asking for. This is I'm not asking for that. But what I do want to know is when we genetically survey animals and we find something that is 69%, is that high enough to be considered a special animal? And should we do something with it? Should we should put a collar on it? We should maybe give it a collar that's orange and tell everyone, don't shoot the orange collar.
Yeah.
If it's not an orange collar, you do what you normally do. But if it's orange, don't shoot it. And then maybe follow up with some other planning, but certainly working in a very kind of modern day, flexible dynamic of community support, but also supporting an endangered species that is still essentially living where it was 40 years ago.
I mean, is there a widely accepted theory about why coyotes, why wolves, or at least why individuals or small populations might decide to crossbreed just in general? Or is there something they might find desirable about crossbreeding that researchers can really perceive? In one of your articles, I think you mentioned anthropogenetic mortality, habitat degradation, translocation, et cetera. Can you sort of unpack how those things lead to the desire to crossbreed?
Yes. I think the very sort of, if I had to distill this into a single answer or a single variable, in working with gray wolves and knowing the work that people have done with red wolves, when you have animals that live in a pack, have reliance upon the group structure, and pick a mate, they have a mate that they are supposedly bonded with and will have several seasons of offspring with. When that mate is lost, possibly due through hunting or collisions with cars, if they've lost their mate and the breeding season is coming up, it's really easy to imagine that the drive to reproduce is very strong. And if their mates are conspecific, so if a red wolf is looking for another red wolf and it's difficult to find, but there's this really handsome coyote strutting around, that's going to fill the immediate need. Gray wolves, I'm not suggesting they hybridize, but when you break up a breeding pair, there can be new individuals that kind of usurp the pack structure and all of a sudden are now the leadership when they previously weren't, or that pack may not see reproduction that year. So there's different responses, but that is a very dramatic change that could easily result in a hybrid mating event. Other options are far more complex, but the breaking up of a breeding pair is very visible, and you can see that within a season should you be watching for it.
Wow, there's so much there to think about. So, in your research and your expertise, do you believe that the breeding program for the red wolf and the findings that you're getting with these coyotes from Louisiana, do you think that this is more viable just on the research end as opposed to the conservation end? Or do you think that there's some way, like Stephen was saying before, that certain individuals that have these high markers of red wolf will be able to reintegrate back in and help alleviate the stress of the animals in that breeding program?
That is a great question. My personal hopes and dreams is that we can use this population in a way that will establish an independent breeding population. There is no inkling in my mind that it's feasible to move an animal from Louisiana up to a captive breeding facility. Those are pedigrees that are managed, and they're very, very careful. And they should be concerned to not let something else in that might disrupt what they have managed. And they're doing a great job, but they have limited animals. So there's a possible idea of, in a breeding program, some animals are prioritized for up next to be in the reproduction pot of, the lottery, and those that might be lower prioritized for maybe age or other health reasons. I think that the suggestion of any of those lower priority animals, if they could be moved back into the wild, now they've been raised in captivity, so even though all of the facilities try really hard to keep them from acclimating to humans, there is an idea that if they could be released back into some protected property where a red wolf can meet up with a 69% red wolf animal, that we might see more of this conspecific mating happening, that animals with higher red wolf content will weed through the coyotes and find another one that's similar to them. And perhaps this could restore some red wolves naturally on their own, let them make these decisions out in the wild.
And how often is the black phase observed in red wolves or coyote populations?
We saw a black phase. Joey captured one this past spring, and it's hot off the presses. I'm still trying to manage all of my different analyses to hone in on that one to see if it has red wolf genetics. So it was a melanistic animal, so beautiful. I want to make sure it wasn't a dog mix of some sort, but it was a beautiful animal. And I think we have pictures up on the Gulf Coast Canine Project website. There's our photographer, Amy Schutt, does such a great job. And those pictures, there's a gallery. So I think there's some pictures there, if not a video of Joey handling the animal. But that's the first one that we've seen in the 2 seasons. But there's always reports. In Pennsylvania, when I do coyote work, every year there's mention of one melanistic. So it's rare, but they're out there.
They're out there, yeah.
Okay, this may be a stupid question, but does the fact that most of this black phase seem to be observed in North America, does that mean that, does that say that this black phase originated in North America? And secondly, if dogs were domesticated some 40,000 years ago, how does someone in your field, what methods do you use to make a clear distinction that the black phase originated in dogs and not in wolves?
Yes, good questions. They might have a long answer, so I'll try to keep them concise. The first question about melanism in North America. Melanistic animals are found in Europe as well. So it's not a limit to North America, but there has been sort of a misunderstanding that when you see a melanistic animal, that it has to be a dog hybrid of some sort.
Right.
And that's not true.
Interesting.
And now I think that leads me into your next question about how do you know sort of when and where it happened? The hopefully short answer is when we find the mutation, so that's a whole nother effort of finding the mutation that gives the animal melanism, we then want to look in the neighborhood around the mutation because there are genetic signatures that we could look for to tell us if it's old, how old, if it's new, and potentially where it came from. So by looking at the neighboring or the flanking sequence, we're able to see that this is a relatively old mutation, and the break at which that flanking sequence happens tells us something about where that mutation came from. Did it come from a wolf years and years and years ago, or did it come from a dog? So through some of these molecular signatures, the age has been estimated, and that was about the 14,000 years ago. And then Reina's work in looking at that, so it's called the haplotype, not only the mutation, but the things around it. When we look at the haplotype and sequence that across North America, or you can sequence it across Europe, you can then use population genetic theory to understand where is it the newest in a population and where is it the oldest. And that helps us know on a geographic scale, maybe where did it enter the continent, how did it flow and spread all across, all the way down from Northwest Territories in Canada down to Florida.
So is it possible then that this animal could have come over, I don't even know, and this is me not remembering, I guess, prehistoric history, when the land bridge was still there between the two continents. So it could feasibly be that an animal with that pigmentation came across the land bridge, landed in North America, the land bridge separates and then it's there basically. So okay.
Yes, that's right.
Yeah.
So dogs are much older than this mutation. So the mutation happened in dogs, and could spread across dogs, however people were breeding them and taking them with them. Right. And as humans migrated, they take their dogs, and those dogs bred with local wolves, local coyotes, wherever you are, and that's how they would have been introduced into each of those species.
Yeah. Speaking of that, how do domestic dog genes manifest themselves in coyotes usually, if not via the color of their fur?
Yeah. It's a good question. I think that a lot of the odd looking coyotes that people suspect is a dog hybrid. I've heard stories, everything from the coat color to it being like a wired-haired dog, a wired-haired dog running around with coyotes, but yet, it might test to be part coyote. So I'm sure there's more hidden nuance, and I'm never really out looking at coyotes running wild. I would love to. I mean, very rarely have I that opportunity, but I think people are pretty good about picking up on that looks different. And I don't know why it looks different or how, but
yeah. It seems to be good that it's the science that you're in seems to be that it's becoming more communal and that people are reaching out to you, so you don't have to necessarily be out in the field, which I think is interesting. That people are, it seems more of a regular occurrence that people are throwing up trail cams. They see stuff in the woods, so they want to get a picture of it, and so they look at it, they think it's strange or different, and therefore they end up finding someone as yourself. It's just an interesting facet to life now, that as opposed to where people would see things and you couldn't get a record of it, now everybody gets a record of everything, and so you have more assets at your disposal, basically.
Yes. I think there's a couple things to that. First of all, everyone has, technology at their fingertips all hours of the day. And citizen science has become a really strong movement in the past 10 years, I would say. So people are actively encouraged to engage. I also think there's perhaps a new generation of scientists who are more open to having these conversations. And not everyone in my generation or the previous generation are open to it, but I find it incredibly valuable. I also want people to know what a scientist looks like. What do they sound like? That, in my opinion, I feel like I can talk to people and connect to people. And I think, especially through COVID, there's a lot of frustration and mistrust in science. But that's also like science evolves and our knowledge evolves. So I do want people I want to push them to think about why something we knew 10 years ago is different from what we know now. And how did we learn that new bit that changed the way we think? And I want to help talk to people about that. We just had a town hall in Galveston last week to talk to the community about coyotes. Because I don't want to just be a name that seems to come in from New Jersey and does something with coyotes and then disappears. And I think a lot of my colleagues feel the same way, that we are reliant upon the community to, at least some of them, support what we're doing, support the vision, and play along, not just try to kill every wildlife that you encounter, but think about coexistence and know who you talk to. You can talk to the animal control, Josh Henderson in Galveston, and you can email me and Kristen and Joey, and we're pretty much going to respond or call you back. And I like that accessibility. We shouldn't be isolated. I mean, I'm an educator. I want people to come to me if they have a question.
Yeah, the open door policy is really great, and it's really the main drive of what we do here on the podcast too, is to have as many people at the table as possible. and as many voices from different angles, just because otherwise, you know, it wouldn't work. None of this would really work. Your research, you know, everybody's research would suffer, and it would just, you know, we'd all be at combating ends of the spectrum, and that wouldn't really work out that well.
I agree.
Yeah. With the VonHoldt lab, because I want to touch on this before we wrap, are you doing a lot of, so everything that Stephen and I have discussed with you today are you working on a lot of these projects in the VonHoldt lab? Is it strictly I know I laid out a couple different things before we started, but are you guys working on the red wolf ghost alleles there? Are you working on the ecological epigenetics? Is that the basis of the VonHoldt lab and what you guys are doing there?
John, yes, but it's my research. So for whatever people think, I go in, I just did lab work this morning. I prepare my samples, I sequence them, and I analyze it and write a paper. My students and postdocs have their own projects. So, for example, I do have a graduate student, Dritty, who is working on the epigenetics of hypersocial mutations in dogs, and she's doing a phenomenal job. She's looking at proteins and how chromosomes fold. It's really great, but she's doing all of that. I'm here to give her guidance. I have a postdoc, Stephen. He works on northern elephant seals, his own research program, and I'm here for moral support and anything professional that can help him achieve his dreams. And then I have a fresh new, I guess she finished her first year, grad student, Stavi. She's going to work on marmots and elephant seals. So
marmots,
yeah. So the VonHoldt lab collectively, we ask these same kinds of questions, but I maintain my own active research program. And that's what everything we've talked about today is what I work on. And it is mostly separate from what the rest of my group does. But we all talk about evolution and natural selection and gene regulation and all of that, how it fits into ecology. Yeah, it's busy.
That's so cool. I mean, just really, what's the process of getting your own lab? I always wonder what that's like. There are obviously others that have, you know, their own lab, but you have your own. What is it like to have that and to get that, just to be able, like you said, to go in and do research in the morning and then come and do a podcast in the afternoon and then maybe going back and do research
I'm getting
again.
beer after this.
How cool is
that? I guess everyone talks about blood, sweat, and tears. Most of me, there was sweat. I had tears once, but that was Bob Wayne's fault. I will call him out on that. My advisor. Bob, I know. Most of this is I love it. And I remember taking my first genetics class, and my father had always helped me in high school because math was sometimes frustrating. And he always helped me and supported me and let me do it after a little bit of guidance. And I think that really gave me the love of quantitative efforts. I saw that there was an animal at some point. and as I took my first genetics class in my senior year of college, during my psychology degree, I was taking genetics. I'm like, I love this. I knew that I needed to do something with genetics in my life. It was problem solving math is involved, but in a very non-math way of thinking about genes and things that are invisible generally. So I worked a lot, and everyone works a lot. I don't know why I got this job. My colleagues could tell you that they believed in me, and when I did the job interview, they saw something, and I can only thank them for that. But I believe that I would have had a job at some point somewhere. If it wasn't here, it would be somewhere else. But just trying to keeping with my vision and liking what I do, I think is so important. I want to be at work 10 hours a day. I want to work on the weekends. I find joy in writing and analyzing and emailing colleagues to tell them, look what I just discovered. Like, this now leads me to 5 other questions. So it's an endless cycle, but it's so satisfying. And I get to like discover something on my computer screen before anyone else sees it. And that happens all the time.
That's incredibly important.
Yeah.
That's really cool.
Fun.
It's really cool. Yeah, it is.
I was just asked by a student the other day. I was asked about, how about work-life balance? Like, they wanted to understand work-life balance. And like, well, I'm probably not, I have no answer. I'm not a good person to ask because I love work and work is not a problem. So I just work. Balance. What's balance? But it makes me happy. And it's even though it stresses me out, it also relaxes me. So I'm a unique person, I'm told.
Yeah, it's awesome.
Yeah, that's a good take though. And listen, all the congratulations on that for just having that space for yourself to be able to do the work that you do, for having this discussion with us and really diving into all these genomics, as I say it right, finally a thing, and these epigenetics. This was incredibly fascinating for me to really understand the work that you do and what's to come, because obviously this is a never-ending story or saga of genetics and things in these canines. My last question for you, Bridgett, is when you hear the word wolf, what is the thing that comes to your mind?
That comes to my mind. Well, I sit here at my computer and behind it is a picture of a gray wolf from the Swan Lake Pack in Yellowstone. So this is burned into my image, my brain, because it's what when I look away from my screen, I see that. And it was also gifted to me when I graduated with my PhD, and it was taken by a good friend of mine, Dan Staler from Yellowstone National Park. He's a wolf biologist there. We got our PhDs together. So I think that's what I think of. Although coyotes also enter my mind all the time. And I know you said wolf, and a coyote's not a wolf, but it's one of those underdogs that everyone's like, oh, there's a coyote. Everyone like, yeah, I don't know. It's not often do they get a positive word. And I'm a big fan of coyote. So I think of that too, I think.
That's good. They really are. Yeah. This is
they're an underdog.
They are. This is our second, I think, coyote-esque podcast, which is good. And I'm glad we ran the spectrum of wolf and coyote again, because they're similar in a way, and they roam the landscape together. So clearly they have found a balance and been able to coexist with each other. So Bridgett, just tell everyone where they can find you, both so on Instagram maybe, where they can look up stuff for the VonHoldt Lab, any of your research work. Just give everybody the details on where they can look at all this work that you're doing.
Oh, goodness. Okay. So I am on Instagram. I had to just look up my handle. I am called The Beeps, and I don't post frequently, but you'll get pictures of my cat, my dog, wildlife, and sometimes food. Oh, my twin sister. If you just Google Princeton and VonHoldt, should be the first one that pops up. But I do run a project called North American Canine Ancestry Project. You can Google that, and that will show you all of the genetic ancestry work that we conduct. I'm also part of the Gulf Coast Canine Project. And if you Google that, or just type in Gulf Coast Canine Project, you'll see the work that we're doing on the red wolf fancy coyotes along the Gulf Coast.
Awesome. Yeah, no, I found all those. I think Steven's looking at them right now. So anybody who's listening, I'm going to have those websites plus the article for the red wolf ghost gene, and also the paper that you were mentioning, Bridgett, earlier.
Yeah, I'm going to email you.
Yes, email me the paper. So you guys are going to have a lot of reading. Those of you that have listened to this, you're going to have reading homework too, so you guys can understand what's going on. Bridgett VonHoldt, this is such a pleasure. Thank you so much for joining Stephen and I and just giving us all the information.
Yeah, thank you so much.
We really appreciate it. Thanks so much.
And I'm so honored to join the list of names that you guys have too. I looked at that and I'm so honored. That's so great. I have achieved it. I'm done. File my retirement.
That's it. She's got a retirement. She reached the mountaintop and now she can go have a drink.
Yeah.
Exactly. Yes. Oh my God, she can give in rotation. Maybe one of us can take a day off one day. We're not there yet. Oh my goodness. Thank you, Bridgett, so much. House to all of you out there, and Stephen, I'll be with you next time. Bye, everybody.
Bye-bye.
Looking to support Wolf Connection or sponsor one of the wolves in our pack? Just go to wolfconnection.org, click on the donate tab, and find out more information.